Hello students. The Belgian chemist Helmont once conducted an experiment. He cultivated a willow tree in a known quantity of soil. After 5 years, he found that the willow tree had gained 74.47 kilograms in weight, while the soil had lost only 0.06 kilograms. From this, he deduced that the tree had transformed water into its own substance. The process he demonstrated is actually the process I am going to discuss with you today: photosynthesis. I will explain it mainly from 3 aspects: an overview of photosynthesis, the process of photosynthesis and energy transformation, and photosynthesis in crop production. Overview of photosynthesis. 1. The concept of photosynthesy. The narrow concept refers to the process by which green plants use light energy to synthesize CO2 and water into organic matter while releasing oxygen. The broad concept refers to the process by which phototrophic organisms use light energy to synthesize CO2 into organic matter. This is because photosynthetic bacteria can also perform photosynthesis. 2. The site of photosynthesis is the chloroplast. Thylakoids within chloroplasts contain pigments for absorbing, transferring, and converting light energy, primarily carotenoids and chlorophylls. 3. The significance of photosynthesis. The process and energy transformation of photosynthesis. According to the requirement for light, photosynthesis can be broadly divided into the light reaction and the dark reaction. The light reaction occurs only in the presence of light and takes place on the thylakoid membrane, also known as the photosynthetic membrane. The dark reaction occurs in the absence of light, or in light, and takes place in the chloroplast stroma. Photosynthesis can be roughly divided into 3 steps: the primary reaction, electron transport and photophosphorylation, and carbon assimilation. The first and second steps belong to the light reaction, and the third step belongs to the dark reaction. When the primary reaction refers to the process from the excitation of photosynthetic pigment molecules by light to the initiation of the first photochemical reaction. It includes the entire process of light energy absorption, transfer and conversion into electrical energy. 2. Electron transport and photophosphorylation. The high-energy electrons generated by the primary reaction are transferred through a series of electron carriers, eventually reducing the oxidized form NADP to NADPH. Accompanying this process, the photolysis of H2O establishes a transmembrane proton motive force, initiates photophosphorylation and forms ATP. Thus, electrical energy is converted into active chemical energy. In this way, the energy captured by photosynthetic pigments is stored in NADPH and ATP, forming the assimilatory power for the dark reaction to assimilate CO2. The photosynthetic electron transport chain is the complete electron transport pathway, composed of multiple electron carriers located on the photosynthetic membrane. The currently widely accepted model is the Z-scheme proposed by Hill and others. That is, electron transport is completed through the coordinated, sequential action of 2 photosystems, PSI and PSII. Photophosphorylation refers to the process in which chloroplasts synthesize ATP from ADP and inorganic phosphate, Pi, under light, generating high-energy phosphate bonds. Electron transport is coupled with photophosphorylation via ATP synthase. 3. Carbon assimilation. CO2 assimilation, or the dark reaction, abbreviated as carbon assimilation, refers to the process by which plants use the assimilatory power formed in the light reaction to fix and reduce CO2, thereby converting inorganic substances into organic matter and transforming the active chemical energy formed in the light reaction into stable chemical energy. Carbon assimilation in higher plants includes the C3 pathway, the C4 pathway, and the CAM pathway, crassulation acid metabolism pathway. 1. The C3 pathway. This is the most fundamental CO2 assimilation pathway. The C3 pathway was discovered by Calvin and others in the 1950s, so it is also called the Calvin cycle, since the acceptor for carbon dioxide in this cycle is a pentose, ribulose 1,5-bisphosphate. It is also known as the reductive pentose phosphate pathway, RPPP. Most plants possess only the C3 pathway. Such plants are called C3 plants, e.g. rice, wheat, etc. The biochemical process of the C3 pathway can be roughly divided into 3 stages: carboxylation, reduction, and regeneration. 1. Carboxylation stage. The carboxylation stage refers to the stage in which CO2 is fixed by a CO2 receptor to form carboxylic acids. Ribulose 1,5-bisphosphate RuBP is the CO2 receptor, which, under the catalysis of ribulose bisphosphate carboxylase, Rubisco, combines with CO2 to form 2 molecules of 3-phosphoglyceric acid, 3-PGA. 2. Reduction stage. 3-phosphoglycerate is catalyzed by 3-phosphoglycerate kinase to form 1,3-bisphosphoglycerate, which is then reduced by glyceraldehyde 3-phosphate dehydrogenase to glyceraldehyde 3-phosphate, GAP. This forms the first 3-carbon sugar and represents the CO2 reduction stage. This is the stage in the Calvin cycle that consumes assimilatory power, ATP and NADPH, and also serves as the junction between the light and dark reactions. When CO2 is reduced to GAP, the energy storage process of photosynthesis is completed. GAP can either form starch within the chloroplast, be transported out of the chloroplast to synthesize sucrose in the cytoplasm, or remain in the cycle to enter the third stage, the regeneration stage. 3. Regeneration stage. The regeneration stage is the stage for regenerating the CO2 receptor, ribulose 1,5-bisphosphate, RuBP. It is the process by which GAP undergoes a series of transformations to reform the CO2 receptor RuBP. This stage also requires ATP generated by the light reaction to provide energy. For the Calvin cycle to continue, the carbon dioxide receptor RuBP must be continuously regenerated. The overall equation of the C3 pathway shows that in the entire Calvin cycle, for every molecule of carbon dioxide fixed and reduced, 3 molecules of ATP and 2 molecules of NADPH are consumed. 2. The C4 cycle. The C4 cycle occurs in C4 plants, such as corn and sugarcane. The C4 cycle consists of the following 3 steps. 1. Phosphoenol pyruvate, PEP, carboxylation reaction. PEP is carboxylate to form oxaloacetate. Oxaloacetate is then converted to malate and aspartate, C4 acids. 2. The CO2 produced by the decarboxylation of C4 acids enters the Calvin cycle. 3. The pyruvate produced by the C4 acid decarboxylation reaction is catalyzed by pyruvate phosphate dikinase to regenerate the CO2 receptor PEP, allowing the cycle to continue. 3. The CAM, crassylation acid metabolism, pathway. This occurs in plants such as cacti and bromeliads. It includes 2 steps. 1. At night, CO2 is fixed to produce malic acid, which is stored in vacuoles. 2. During the day, organic acids are decarboxylated, and the released CO2 participates in the Calvin cycle. 3. Photosynthesis and crop production. 1. Light energy utilization efficiency of plants. Light energy utilization efficiency refers to the percentage of the inert stored in photosynthetic products relative to the total solar radiant energy reaching the Earth's surface. Reasons for low light energy utilization include 1. Light leakage loss occurs during the early growth stages of crops when planting is too sparse. 2. Waste caused by light saturation occurs under intense light. 3. Inappropriate environmental conditions and improper cultivation management. 4. Other factors such as environmental pollution. 2. Ways to increase crop yield. 1. Enhance photosynthetic capacity. This requires rational regulation and improvement of temperature, light, water, and fertilizer conditions. 2. Increase photosynthetic area. Leaf area index, LAI, is often used to represent photosynthetic area. Measures such as reasonable dense planting and altering plant architecture can increase photosynthetic area. 3. Prolong photosynthetic time. This includes the following. 1. Reasonable intercropping and relay cropping to increase the multiple cropping index. 2. Selecting late maturing varieties and using methods like seedling transplanting to extend the growth period. 3. Using bioluminescent lamps and fluorescent lamps as artificial light sources for supplementary lighting. 4. Using photorespiration inhibitors to reduce photorespiration and preventing diseases, pests, and weeds to reduce organic matter consumption. 5. Coordinating the relationship among source, flow, and sink to improve the economic coefficient.